I think there's sort of two flavors of biotech or health tech startups. Either you start from sort of this deep expertise on the biology side, or you start from the deep expertise on the technology side. I am just a thoroughbred biology nerd. In 2015, I left academic research and I joined Color. It is a company that's really focused at the time on germline genetic testing, making genetics and genomics more accessible to all patients.
How do you apply that kind of startup mentality to healthcare?
I have become a firm believer in sort of that operating model, which is to move with urgency and to sort of bias towards action. What you learn when you're a startup is that you have no other advantage but speed. The product will not succeed as a product if you don't embrace the technology side.
What does it take to build something new in healthcare beyond the technology? How do you bring together the team, the culture, and the mindset to pull it all off? That's the question running through today's episode with Alicia Zhou, the CEO of the Cancer Research Institute. Today kicks off part one of our two-part conversation. Alicia started her career as a bench scientist and made the jump most scientists don't make. 9.5 years as Chief Science Officer of Color Health, one of Bay Area's leading genomics companies. She now leads CRI, a nonprofit at the very center of cancer immunology research and funding. Today, Alicia breaks down what really worked at Color Health and why most health tech companies stumble in getting technologists and scientists to collaborate. We talk about every startup's only real advantage, speed, and what it actually means to hire for the ability to thrive in environments of uncertainty rather than just competence. Finally, we discuss why just a brilliant innovation does not automatically mean you have a viable product. If you're building at the crossroads of science and technology, you don't want to miss these insights. Let's get into it. So Alicia, you have a very interesting story. When I was looking you up, you're a Taekwondo champion and you are a tech executive, and then you kind of landed in this role at the Cancer Research Institute.
Yeah,
We just start by just sharing with the audience like your story and how you got to where
sure.
you are today.
I would say that the best way to describe me is I am just a thoroughbred biology nerd. That's sort of my North Star. And it's something that I've always really enjoyed is thinking about how to make impact in the world with biology. And so I actually started out being a bench scientist. I did academic research in the field of cancer biology. I was really lucky to train with some of the greats in that field. I went to school at MIT and got my PhD from Harvard, and I had the opportunity to work with Bob Weinberg, who's a legend in the space of oncogenes and cancer biology. And while I was doing my PhD, I also had this opportunity to work at the Broad Institute that was building this massive data set as part of the NIH's Cancer Genome Atlas program. And it really sort of changed the way I thought about how you would do bench science and realized that data could really propel that. I came to the West Coast to do my postdoc at UCSF, where I trained with Andre Goga and Mike Bishop. Mike just passed this weekend. I'm very sad about that. But he was really formative in my career. And being in the Bay Area, I think there's sort of this obligation to consider becoming an entrepreneur. It was not in my background. It certainly wasn't. I don't think anybody would have bet that I was going to join a startup, but I did. So in 2015, I left academic research and I joined Color. It was called Color Genomics at the time. Today it's called Color Health. It is a company that's really focused at the time on germline genetic testing, making genetics and genomics more accessible to all patients. And then I had the privilege to become their chief science officer, and I was there for 9.5 years. And that is a company that's really now playing at the intersection of technology and healthcare, and really thinking about how do you bring products into the hands of patients through the lens of employers and doctors to be able to maximize for patient outcomes, but in a way that sort of helps the ecosystem of healthcare. So it's really interesting. I was there for 9.5 years. And then about 1.5 years ago, I was recruited to become the CEO here at the Cancer Research Institute, which is a nonprofit organization really focused on how do we fund basic science research for cancer immunology, which has become this really interesting field that has become this fourth pillar of cancer treatment. Today, immunotherapy has become this ability for some patients to get cures There are some tumor types now where we know immunotherapy is first line. And how do we make it so that immunotherapy can become useful for all cancer types is one of the things that we're really focused on.
It's really special to kind of meet somebody that has kind of gone through, the whole evolution from being a bench scientist, being a tech executive at, one of the San
Mhm.
Francisco Darlings, and then, going on to the nonprofit side, funding research. I think it's a really interesting vantage point that I think you can offer, our audience. I wanted to kind of hit on, your experience at Color Health first, right? Like, you spent nearly a decade there, building startup world. I'm curious, like, how do you apply that kind of startup mentality to healthcare? And, what are some of the key learnings that we can kind of share with our audience?
Yeah, it's so interesting. So I think what was really important and special about Color was that it was a technology company first, and then it understood the value of experts in the field. I think there's sort of two flavors of biotech or health tech startups. Either you start from sort of this deep expertise on the biology side, or you start from the deep expertise on the technology side, and then you're in this. You have to very quickly acquire the right talent on the other side of the aisle to ensure that you have that sort of shared knowledge. I think what Color did really well from the very beginning was that they had experts on both sides immediately,
Mm,
and they really just engendered this feeling of collaboration, and it came from this sort of humility to say, "Look, I really know the things that I really know, and I know nothing about the things that you know. How do we work together so that we can share our knowledge and our expertise to build a product that's really going to really change the way people get access to healthcare, right?" And so I think that's one of the things. I think wherever you're starting in the startup journey, if you are tech heavy today or if you're biology heavy today, you must add somebody from the other side, and then from there, I would say, this is more advice to the biologists in the world like me. As a biologist, I think we scientists tend to underestimate the value of the technology. And so we come in with our like big brains and say like, oh, but clearly it's because of the biology that this product could exist. And like, yes, that is probably true, but the product will not succeed as a product if you don't embrace the technology side or understand how to leverage the technology. And so you also, both sides, you have to bring the humility to be able to collaborate with the other person on the other side. And I think that's something, if you can't figure that out, it just creates friction on the team and it creates a lot of inefficiency. And as a startup, you really can't afford that. You have to be as efficient as possible. And so these are sort of the things I think about.
I think speed is such an important thing for startups. And
Yeah.
one of the things that you touched upon was, how do you actually get this collaboration between technologists and the
Mhm.
scientists? And I feel like at Color, and you guys have done it so well, especially kind of, I don't want to call it pivoting, but like having
Mhm.
three distinct evolutions of your business. Can you just touch a little bit more about like how do you actually solve the problem of urgency and driving urgency and getting that collaboration down at an
Yeah.
organization, especially like moving from the startup world to
Yeah.
now CRI, where I'm sure you're trying to instill some of those operating principles as well.
Yeah, absolutely. I mean, I have become a firm believer in sort of that operating model, which is to move with urgency and to sort of bias towards action. It's something that was very much instilled in me at Color, and I immediately saw the advantage that it had. At the end of the day, what you learn when you're a startup is that you have no other advantage but speed, right? At the end of the day, the incumbent has more cash, more brand, more team, and way more runway than you do. So you're kind of set up to fail, but what you have is agility and speed that they don't. And so if you sacrifice your speed, then you have nothing. And so as a startup, actually, it becomes a really driving principle, which is that, okay, we don't have nearly as many resources, we have to be more scrappy, but we can pivot much faster. We can move much more quickly. And so I think one of the things that you kind of learn is we talk a lot about uncertainty, and there's been a ton of uncertainty over the last, let's call it, decade, between this massive global pandemic that disrupted everything, to how AI technology is changing the way we work, to all the crazy things that are happening on the funding side for science, right? And so there's always this like unprecedented time. Well, we've been in an unprecedented time for like an unprecedented amount of time, you know what I mean? So at some point you actually kind of have to say this is the norm, and then then it becomes this thing of understanding actually, how do you embrace uncertainty? I feel like somehow we've decided that uncertainty is bad. It's like a bad thing. And actually, the way I think about it is uncertainty is the time when opportunities become available for disruption, right? And if you're a startup and your job is to disrupt, you actually want to lean into the uncertainty. You actually want to take advantage of the uncertainty because the incumbent in this moment is going to take longer to make a decision and have a harder time moving than you, the smaller player. And so for that reason, I think as a startup, and what I say to the folks at CRI and the advice that I give to people is see the opportunity in uncertainty. Instead of feeling like this is unstable, actually think of it as this is a ripe opportunity for disruption. How do you find the right leverage to create the product or the program, in our case, that's going to really shift the dynamics of the market, right? And that's something that when you think of it that way, then you also think about the people you're hiring. One of the things that I've really learned over the years is that one, of course, competency is a must-have. You must be good at your job. That's sort of table stakes. But there are people, especially people coming from my side of the world, who are super, super competent, but they shy away from the speed and the uncertainty. And so then this idea of uncertainty becomes really difficult for them. One thing that I've learned is you have to hire people who can metabolize uncertainty very well. Meaning like uncertainty happens and they're like, no problem. Like I eat that for lunch. Like, let's go. Right. And like, and ideally they even enjoy it. Right. And that they like that sort of unknown. They like to work in these underdefined, underspecified parameters. That type of person, who of course is also meets the bar of competence, that person is really going to help your company position itself quickly into places where it needs to go. And ultimately, that's how you pivot quickly.
I love that so much. Basically, if speed is the only thing that you have to be able to embrace uncertainty, and therefore you need to find people that are able to metabolize the uncertainty. Said like a true biologist, by the way.
Yeah, that's how I think about the world.
You have to metabolize it, and
Mhm.
I think that's so true. And I think we're starting to see that in the work that you're doing as well at
Mhm.
CRI. I know
Mhm.
CRI has been around for a really, really long time, but embracing this disruption, one of their major initiatives I know is the discovery engine that
Yeah.
you guys are working on. And it's really kind of like a novel thing that the industry has never seen before.
Mhm.
Can you kind of tell us about that? And,
Sure.
what is the problem that you guys are trying to solve with the discovery engine?
Sure. It's so interesting. For me, multiple times in my career, I have come to realize that biology is only supercharged by data. And so there's sort of two places I've seen that before. One was with the TCGA, the Cancer Genome Atlas, which I mentioned. I watched TCGA get built when I was in grad school. The Cancer Genome Atlas is this huge database that was built by the NIH that was a catalog of all these patient tumor samples that were then subsequently sequenced at a pretty high depth for many of the genes that we believed were driving oncogenesis or driving the cancer. It was done now 20 something years ago. And so at the time, sequencing was incredibly expensive. We weren't going to be able to do whole genomes or even whole exomes on all of these samples. And so what was happening is these samples are being sequenced across these multiple hundreds of genes panels, really helping us understand, what are the driver mutations and sort of the genetic repertoires that are activated or silenced in these different tumor types. And it changed the way you did biology suddenly, right? Where you didn't have to kind of make yourself believe that that your gene was a becoming a protein that was activating this other protein. You weren't doing this sort of stepwise mapping of biological pathways. You were actually using the existing data to kind of impute backwards on what are the pathways that are most disrupted, what genes tend to be overexpressed or silenced together. And it actually gave you an insight about how these genes potentially talk to one another. And that was really interesting. I think that really started this, I would say, sea change within biology, where We went from doing hypothesis first research to doing data first research. And I
Mm.
think data first doesn't mean you don't have a hypothesis. It just means that the data informs your hypothesis. And so, when I was at Color, one of the things that I got to be involved in was the NIH's All of Us Research Program, which is this large initiative to sequence 1 million people's whole genomes and then put them into an open access database with EHR data and harmonize that data across. And that is an initiative that today has about 800,000 genomes generated, and those data are available to researchers to use. And that was also just a really powerful testament to, if you can get rich harmonized data, it can really change the way we can do research. So I kind of came into CRI with this idea of, okay, we know that data supercharges biology, and coming in 2025, like, this is the era of AI. Like, whatever is going to happen, AI is only going to supercharge it even more. So how do we think about, how do you prepare the field of cancer immunology for this sort of data AI enabled future? And one of the things I was thinking about is, it really comes down to having a foundation of, or a truth set of data that are going to inform the foundation models that are going to be built on top. And so we know the compute is there, right? We see it, we use it every day. The best examples, of course, are in these large language models that we use, like ChatGPT and Gemini. We know the foundation models have already changed the way we've done biology and protein folding, for example, with AlphaFold and DeepMind. How is this kind of technology, how are foundation models going to change cancer immunology? My thesis is that we should create the truth set of data, the ground truth set of data that will help train those foundation models. And when it comes to immunology, it's really complicated because we have so many different cell types. They are all talking to each other. It's a dynamic system. And the thing about the immune system is that it's most interesting to be studied when it's being perturbed, right? Like, your immune system, for the most part, is always there in the background. It's keeping you alive and keeping you healthy. But really, the immune system is activated when when a pathogen comes, right? When you have an infection. And in the case of cancer immunology, the immune system is activated when it's treated and when it's trying to interact with the cancer. And so one of the things I realized is that if we're going to make this data set, it couldn't be a static data set, and needed to be something where we were studying biology, a system while it's being perturbed, and looking at what's happening in over time and in space. How are cells talking to each other? So ultimately, that's how the Discovery Engine got born. It's about studying basic cell biology and immunology as a perturbation is happening, looking at cells in time and in space. And that's really what the CRI Discovery Engine is.
Wow. It's amazing, and it's probably like really like the first of its kind. Like, you know, as someone that is, that has this thesis, is seeing this data set get
Mm.
built, I'm curious, like, what do you think is going to come out of it? Right? Like,
Mhm.
I think you obviously have some hypothesis of like what this is going to eventually lead to.
Yeah.
What are
Yeah.
you most excited about? And, what do you think is in there that is, this is really the key to unlocking?
Yeah, I mean, I think that we all believe, or at least I believe, that the future of every sector is going to be AI enabled in some way. So the question then is, how is AI going to show up in basic science research and in drug discovery? Um, and so I go back to sort of the adage that we use in science all the time, which is all models are wrong, but some are useful. What we know in biology is we have at our fingertips so many model systems that we use today. Some of those are cell lines that we use. They might be mouse models. It might be human data. But these are all model systems that we are using today. And I don't think there's going to be sort of one model to rule them all. I actually think that a really good biologist is doing the right experiments in the right model to answer their question, and they have all these tools available to them. So to me, then, the bench biologist of the future, the AI-enabled bench biologist, also has a foundation model, an in silico model that they are putting into their tool set. So now they're going to have their mouse models and their cell lines and their humans data, and their foundation model copilot that is helping them think about how the biology works. So this copilot, if it needs to be good, which means that it needs to understand the sort of deep data that is biology. I actually think foundation models are really well positioned for biology because biology, at the end of the day, is very multidimensional. We have genomics data, we have transcriptomics data, we have proteomics data, we have data over time and in space. And so it's a really ripe data set for foundation models. And so the question then is, in this world where that biologist is using a foundation model copilot, we want to make sure that that is a really well-trained model and that it is actually recapitulating biology, right? It needs to be doing, it needs to tell you, make predictions that are true. And so to do that, it needs to be trained up in a way that allows for that reinforcement learning to happen. So what's nice about this discovery engine is that we do bench experiments that will generate data. That data will hopefully be used to train any number of foundation models. But what's nice about it is then if you get a prediction, we can go back to the bench and test it and say like yes or no, so we can reinforce the model training. That's what I'm really thinking about. I hope that there's a world one day where there are dozens of different players in academia, in industry, or just hobbyists who are using the CRI Discovery Engine data to train their own different types of models for their own particular use cases.
Do you think that right now it sounds like the data set is going to be used to train a foundation model that will be kind of packaged into a copilot for a bench scientist to use in their, you know, hypothesis generation or discovery, or even, when they're doing their actual testing, they're able to leverage it to figure out, ask any questions that it wants. Do you think it'll ever be abstracted away from that, where it's like, almost, this might sound like a stupid question, but like, hey, like, right into the foundation model, like,
Mm.
you know, create a cure for this disease, and then it
Yeah.
just automatically does it. Like, do you think we'll ever get there, or do you think it's ultimately like, like, yeah, I'm asking this question just in the context of, um, in AI, there's this, something called the bitter lesson, right? And it's
Mm.
basically talking about how models that ultimately scale with compute will ultimately outperform these kind of hand-tuned models. And it sounds like the process of discovery and the process of science today still has this kind of, um, you know, I would call it like a bottleneck in the context of, reinforcement learning, that you always need a bench scientist to
Mhm.
validate, or to come up with a hypothesis and, keep doing this. Do
Mhm.
you ever see a world where there's just like full end-to-end discovery and testing and, you know, purely through AI, or
Mm.
you think we're really far from that?
I do think we're far from that. And the reason is because we've barely scratched the surface of human knowledge of biology. There's a lot of uncertainty. There's a lot of unknown. We have a lot of places where we really barely understand what's happening from the biology side or the medical side of the world. And there's so much unknown there that if you let a foundation model just imagine all that could be there, like maybe one version of what it imagines is true, but there's just the inability then to reinforce that and say like yes truth versus no hallucination versus like that would be cool but that's not how the world works. I think the thing that's really powerful about foundation models as applied to science is when you have sort of the grounding reality of truth, so like a world that is bound by the laws of physics, for example,
Mhm.
with the completely boundless imagination of a foundation model that is not bound by the laws of physics. This is what's really interesting about AlphaFold by DeepMind, which just won the Nobel Prize 2 years ago for its protein folding algorithm. It actually has two transformers. It has one that is bound by the rules of biology and physics, and has one that is not. And that's super important because if you bound it too early, it's unable to imagine sort of all the possible
Mhm.
ways a protein could fold. But if you never bound it, then it will imagine things that are biologically and physically impossible.
Yeah.
So that's sort of the opportunity for AI. But in order for that to exist, there has to be that grounding piece. And the reality is that we just don't know enough about many parts of biology. that's the half that's missing, not the compute half. It's the half of like, how does it actually work? Like, we don't know.
So how are people contributing to this data set today? Like, is it just through the CRI funded research? Like, do you have partnerships that are with like the large biotechs that are doing like kind of non-competitive research and donating that data? Like, what does the data set look like today? And maybe how can other people contribute or how can other people help contribute to
Yeah.
this true set?
Yeah. So where we are today is we're starting with our academic collaborators. We're working with three PIs. We're working with Ansu Satpathy at Stanford, John Weary at UPenn, and Andrea Scheedinger at Memorial Sloan Kettering. These are three incredible scientists, really well versed in the space of cancer immunology, but also some of the leaders who have pioneered not only some of the discoveries, but also the underlying technology for how we map it. And then what's really important here is we also have a partnership with 10x Genomics, who is really changing the way you can capture this data. So they are the ones who are sort of coming up with these new scalable and affordable techniques to be able to do single cell RNA sequencing at scale, to be doing spatial transcriptomics at scale. So we're starting first with this sort of very controlled environment. I do want to get to a point where we are then making it open for others to contribute their data, or for us to be informed really by where the data is going to be most useful, and tell us, what are the questions or the areas where they want to see more data generated. But for now, we're starting a really controlled environment. This is something that I sort of learned, I think, from working on All of Us and other data projects, is that the quantity of data versus the quality of data is always going to be sort of that push-pull. There are certain questions that you can answer by just having a large quantity of very dirty data, and then you kind of sift through and find, one really interesting feature that is shared across all. You can definitely make insights and products from that. But I think for the purposes of being able to really predict all the different types of biology that occur, I actually am optimizing here for quality of deep data, not necessarily for quantity and breadth.
Yeah, I mean, I think even the way that models work today, I think they're very inefficient at going through unstructured data. But to your point about this tension between like ground truth and like constraints versus imagination, if you structure the data too much, it might be kind of limiting its ability to actually find the needles in the haystack. And so it's a really interesting problem that you guys are solving. Are you guys kind of thinking about it in terms of, you know, you guys are going to define the
Mhm.
repository and the structure of
Mhm.
it, or are you going to have almost like multiple different versions of it
Mhm.
where you have one where it's highly structured based off of a specific use case, and then
Mhm.
you have others where it's almost unstructured, and whoever is leveraging that can determine how they want to structure that data for their use case.
So here's the thing I think a lot about is that biologists have a preconceived structure of how they want to have the data be labeled, and it's a very biased towards biology view, right? Which is like, we think biology works this way, so we want to label and structure the database in
Yeah.
this way. What's super interesting and very worthwhile to consider is that when you go to define your data architecture, and we are at the beginning, so we are at this point, um, I think it's also really important to add in people who are non-biologists, who are more technologists on the foundation model building side, because what you'll realize is that the technologists, they don't subscribe to the religion of biology, right? They're like, whatever, like, I know that's important to you, but like, all I see are a bunch of data. And what they're going to be able to add is what the utility of different features might be for them long term.
Mhm.
And actually, you run the risk of throwing away really useful data that you thought were garbage because a biologist doesn't care, but a technologist really does care. And so I do think it's really important to think about this early on. Sure, you can also just keep it really, really open. It does then you start to ask who your primary user is then, right? Like, if maybe there's a user out there who's just like, give me all the raw data, don't label any of it. Like, I want to do it myself. And that person certainly exists. That person probably is going to do something really interesting, cool, and a bit bespoke. But we also want to be bridging the gap between the person who's super fluent in building models and the biologist that wants to leverage that model. So I do think it's important also to be able to provide data sets that have this sort of basic architecture on top so that we start to have a shared understanding of how we talk about these data, how do we collect these data, how do we make sense of these data. Ultimately, that allows us then to scale. Otherwise, you're going to create these little factions of technologists and biologists who are using it very, very differently. And that's not necessarily going to get you to that sort of shared understanding that you really want.
Yeah, I think it really speaks to like the first thing that we talked about, which was how do you get the scientists and the experts and the technologists and the experts to come together to really come up with a approach that really satisfies or optimizes for both what they're looking for. I think it's so important, right? Because like I think a lot of the data sets out there, they're curated for a very specific use case, but I
Yeah.
think the use case that we're describing is on one hand unbounded, right?
Mhm.
And you have to really have the flexibility or bake the flexibility into the data set such that it's able to support multiple use cases rather than one.
Mhm.
So yeah.
Yeah. Yeah. And it
Uh,
only becomes more complicated as you think about clinical data and human data, which, like, of course, is already being collected in these different EHR systems. And it's all being documented differently depending on what software backbone you're in. So this is the problem, not just this is the problem across the entire healthcare space, and it's worth talking about.
Maybe when you solve it, we can have you back on and you can tell us how you solved
Yeah.
it.
Yeah. Yeah.